Looks like it's 3:01, so without further ado, I will get started. So today's speaker is Dr. David Waddington. He is a Cancer Institute New South Wales Early Care Research Fellow, and he works here at the ImageX Institute. His research translates cutting-edge development in the basic sciences into MRI technology that can personalize radiotherapy treatment. At present, David is working hard on integrating artificial intelligence with MRI for more accurate treatments on new devices known as MRI Linux, which he will talk more about in this lecture. As always, if you want a certificate of attendance, Julia will put her email in the chat, and you can send her an email and she will send you the certificate of attendance. If you have any question, please put them in the chat, and I will ask David whenever there's a convenient moment during the lecture, and otherwise, please keep your question for the end, and you will have a Q&A session where you can ask David the question yourself. Feel free to keep your video off, especially if you're having bandwidth issues. But otherwise, you are also free to leave it on. And I think everybody is muted, but please make sure you stay muted throughout the lecture. And so without further ado, I'll let David take the lead. Go ahead, David.
Hey, Yusuf, thank you for the introduction. And yeah, so here to talk to you about some of the basics of MRI as part of our summer lecture series. And well, let's get started. So I guess the first thing that I want to say is that I think MRI is possibly one of the most complicated imaging modalities. It's also probably one of the richest imaging modalities. But that means that it's a hell of a job to try and teach anyone anything about anything in a short period of time, but I'm going to do my best and hopefully convey some of the fundamentals across in a way that is sufficiently light that you feel like you learned something about quantum physics, but then also means that there's some practical aspects for the clinic or wherever you end up next. So yeah, jumping into it. Well, I think I want to start by saying that, as always, when it comes to us, there's a lot more to humanity, a lot more to physics than maybe it first meets the eye. And one of the things that I think is kind of interesting about MRI is that it takes advantage of sort of an Avogadro's number, 10 to the 26 zeros of natural quantum sensors that already exist inside you. And what are those quantum sensors? Well, they're hydrogen nuclei. And the greatest source of hydrogen nuclei is in water molecules, which, I mean, we're 70% water, so and there's 2 hydrogen nuclei for each water molecule, so that's a lot of hydrogen nuclei. And so what we're going to dig through here is these hydrogen nuclei that are already everywhere. How do we turn that into MRI? It's MRI's magnetic resonance imaging, and what is it about these hydrogen nuclei that allows us to do this? And so I guess we know they're there. So why do we want to use these things? So we know they're everywhere: fat, water, fat and sugars, as well as water. Now the big thing is that they have what we're going to call a spin quantum state that depends to an extreme precision on whatever magnetic fields your body is exposed to. And so for the purposes of this talk, really what I want you to think of all these nuclei as are just these little magnets. Just think of them as little magnets that point in a certain direction and align to the magnetic field that they're exposed to. Now, the thing is, though, that tissues throughout your body have different magnetic fields. So the magnetic field that exists naturally in the fats of your body is different to the magnetic field that might exist in the fluids of your body. So what we do with MRI is we leverage all these differences to get all sorts of information. So, well, how does MRI work? So now we know we have these sensors and we want to try and learn things about the body. We want to take an image. So kind of break it down into a few things. We have this MRI scanner. So got a little cartoon of an image here, and this generates all sorts of magnetic fields that change in space and time. And what we do is we measure the magnetization of all those spins. So all these nuclei. And then what we do is we use our knowledge of the magnetic fields that we've applied to what we call reconstruct, which is a mathematical operation to create an image of the anatomy or the biological function based on the things we know about what we've done. And we get all this rich kind of structure. So here's a typical brain MRI. But, okay, so what is it about these spins? And I guess on this fact, we get down to nuclear spins in a magnetic field are actually sort of fundamentally quantum. And for the purposes of this talk, what that means is that they can be in one of two discrete states. We call these an up state or a down state. And so what that means is if a magnetic field is applied, is a nucleus aligned with the field or is it aligned against the field? And the thing is that these two states, this up state, this down state, will actually switch if you apply radio waves at a very specific frequency. And that frequency depends very precisely on the magnitude of the magnetic field that they're in. So you can apply radio waves, and these nuclei, the nuclei in your body when you're in an MRI scanner will go from up to down, or down to up, or all over the place. And so what we do is we apply these magnetic field, these radio waves, and spin split. Now, how big should the magnetic field that we use in an MRI scanner be? Well, so when we have no field, we see that we have all these spins. These are arrows here. These blue dots are depicting nuclei. They point in all sorts of random directions. And so the answer is that the magnetic field should really be as big as you can make it. And the reason for that is it's quite hard to get these spins to align with the magnetic field. So even when you have a 3 Tesla magnetic field, which is a conventional clinical field strength, it's actually 1 in a million spins that actually aligns with the field. And 3 Tesla is already quite strong. That's the kind of thing that might rip a hammer out of your hand or something else. So we're talking big, big fields. Now, there's going to be 4 important properties here that I want to pay attention to, and that are really important clinically. And so the first of those is what we call T1. And so the T1 is this characteristic microscopic time that it takes for spins to align to a magnetic field. So we apply a magnetic field. How long does it take for those spins to settle into some state aligned with it? Now, the next thing is going to be once those spins are in that magnetic field, something else that's kind of funny is going to happen. If you imagine that if you have this strong magnetic field and you put a magnet next to it, it's kind of going to shake backwards and forwards, let's say. And in practice, what happens is these spins have angular momentum, and that creates something that we refer to as precession. And so what happens is that these spins will, what we call precess or rotate around the magnetic field. And this will happen at a very specific frequency. Now, this rotation, the rotation of the spins around the magnetic field is extremely precise. And so what I mean by that is that I have a spin that's here in my hand, I have a spin that's in my nose, and I have a spin that's in my other hand. If they start off pointing the same direction, they will rotate at precisely the same frequency. They'll go and they'll stay. And we could refer to this as being in phase. Now, what that means is that what this maintenance of phase means is that if we come in and we do all sorts of other bits and pieces, we can actually adjust where that phase is, and we can measure that. But the important bit for now is to remember that this precession frequency, this maintenance phase, is extremely precise, and a row of spins will maintain that phase and point in the same direction. And so that raises the second important parameter of 4 that we're referring to, and that's the T2. And so that's the characteristic time that neighboring spins precess in phase 4. So how long does that one that's in my hand, how long does that spin that's in my nose, and how long is this one over here? How long, if left to themselves, will they point in exactly the same direction? And we call that the T2. It's a measure of that precision. So, and that's so basically what I'm describing here is this T1. We have that time that it takes a spin to reset, to point along the direction of the magnetic field. And then for spins that aren't pointing along the magnetic field, how long do they maintain phase? And typically that's like 100 milliseconds to a second. So the alignment time and the phase memory time. Okay, so that's kind of everything that's important about the nuclei that are in your body. How does an MRI scanner work? How does it get that information out? Well, we have a few things that go into, we really only have 4 things, but a few things that go into an MRI scanner. We have a patient. They're the ones with all the water or the nuclei in them. We have the magnet, and that's the one that polarizes the nuclear spins, that magnetizes them. We then have radio frequency coils. So they're the thing that measures the magnetization. And then we have another thing that we call gradients. And what those gradients do is they can change the magnetic fields in space in different parts of your body. And I'll explain a little bit more about what that means. So to go into a little bit more about, so the radio frequency coils, we have magnetized spins. Really the way that these radio frequency coils is we have a changing magnetic field, so this precession that we spoke about. And for those of you who've done a little bit of physics, and even at HSC level, you might remember Faraday's law or inductive detection. So an MRI scanner works by essentially having a loop of wire with a magnetic field oscillating within it, and that induces a voltage on a coil. It's at some level, it really is that simple. And so to show you what this is one of those coils in practice, underneath all this plastic, you have a bit of wire, and then you have the spins in that person's head going backwards and forwards and inducing a voltage. Now, on top of that, we have these gradient fields. So these gradient fields essentially add a little bit of magnetic field on one side over here and subtract a little bit of magnetic field on the other side over here, as we've got in this picture of a patient in a scanner. And so we've spoken about the idea that these spins point in the same direction and they remain precisely in phase. The frequency at which they are going around also depends precisely on the magnetic field that is applied to them. So these gradient magnetic fields add a little bit of phase over here, and they add a little bit of magnetic field over here, they subtract a little bit over here. And what that means is these things will start spinning at different frequencies. And so essentially what's happening here is that when we take an MRI scan, really what we're doing is we're magnetizing spins, and then we're making them rotate, precess at very slightly different frequencies all over the body, and using these gradient fields and then turning that into an image. That's just an explanation of what I was doing with my fingers. These things are in 3D. The point is more to say that these gradient fields are everywhere in the MRI scanner, and we can really tune each point of the body to have our spins, our nuclei precessing, rotating at a different frequency. Okay, so let's kind of bring all that together. I've thrown an enormous amount at you there. And let's kind of tie it back to the clinic and what's important. So let's talk about in the clinic, we have 2 important parameters that we can choose on an MRI scanner. And they are the repetition time and the echo time. And so essentially what the repetition time means is how often do you measure your spins? And the echo time means how long after you magnetize your spins and start them processing do you measure them? So they're two things that can be tuned in the MRI scanner. And so what we do when we take an MRI scan is we encode the spin magnetization, then we measure the magnetization at this TE, what we call the echo time, and then we wait a repetition time for the spins to repolarize before we do the whole thing over again. And we just repeat that multiple times to get an image of the entire body. And you can summarize all of the processes that these occur in through these things that are called pulse sequence diagrams, but they essentially describe all the different types of images we can get, and by tuning all these different parameters and timing, we can do all sorts of crazy stuff. And I will speak about more about that. Now, this number 4, the repeat, the reason that we have to repeat it many times, and I'm not going to spend a lot of time talking about this, is that essentially what we need to do to construct a complete image of the human body is we need to fill something called K-space. So for those of you without a mathematical background, really don't worry about this. But for those of you that kind of have done a lot of maths, and I know we've got a few people from, say, like a machine learning background and stuff with us this year, really what this is an inverse space. And because we are acquiring things that are in frequencies and have phase, all of our images are actually taken in this inverse space. And so the idea is to fill this inverse space that we call K-space and turn that into an image. And so basically what we find is that as we fill K-space, we can then get all sorts of information. And here we sort of have, for those who've studied these kind of problems, you can see that different parts of the K-space can contain different bits of information about the images that we reconstruct. And in MRI, there are all kinds of crazy ways to combine these things and what we call different trajectories, all sorts of ways to sample our information. Now, again, coming back to what does all this mean in the clinic, though? So remember, again, we have, really, I think, probably should have said this at the start, but if there's one thing I would like you to take away from this, it's some sort of idea of what T1, T2, TR, and TER and why they matter. So that is the T1 and the T2, the times that characterize the nuclear spins in your body. And then the TR and the TE, which are the repetition time and the echo time, which is what we can figure on the MRI scanner. Because across those 4 parameters, by changing those and tuning those, we can get all sorts of different images. So what I've got here is the same brain, but acquired in 2 different images. Now, from a diagnostic perspective, it's enormously useful for working out what tissue is what, that these 2 images look very different. And you can see that different regions of the image have what we call very different contrast, but this bright region here is not bright in this scan here, for example. And so the way that we change regions of the brain are bright and which regions of the brain are dark is by adjusting these TR and TE values on the MRI scanner. And so we can see here, this is just one example, but we have 2 different tissues that have a different, what we call a different T1 time. And what that means is that the magnetization recovers at a different rate. And if we change where we sample these 2, there's either going to be very little difference in terms of, so this is the signal, how bright they are. There's going to be very little difference between the 2 values if we take a long time out here, so it's a long repetition time, or a really big difference if we choose our repetition time to be here. And so that works for, this is for what we call T1 weighted imaging. We tune the repetition time to get this type of scan here. And then we can also do what we call T2 weighted imaging, where we tune the echo time, or the time after that, that time after we start our spins processing that we acquire an image. And so we can also tune times and get different signal intensities between tissues. And then we can do combinations of that give us other sorts of information as well. In this case, just the overall proton density. Okay, so T1, T2, TR, TE. They're the 4 important things. But I also just want to show you some of the things that kind of manifest out of this. What kind of interesting things can we do in the clinic? Well, one of the big ones that you'll see a lot in research papers is functional MRI or fMRI. So what functional MRI does is it measures brain activity by measuring changes in blood flow. So what happens is I show you an image of a cat, or I tell you to think about your parents. And someone will be imaging your brain. And the idea is to work out which part of your brain is responsible for that activity, for the visual activity in that case, or in this demonstration here, move your finger. Which part of your brain is responsible for moving your finger? And what happens is that, and so what was discovered 20 years ago now was that, started being rolled out about 20 years ago now, was the idea that this T2 time that we're talking about in your tissues actually gets longer when you use your brain to do something. So what these images depict here is that within the brain, the signal is going up in a T2 weighted scan because of the because they told you to think of something. And so what happens is that you have some neuron, it goes, I've told you to move your finger, that gets triggered, and then what happens is T2 gets longer. And so this blue, the signal actually goes up, and then actually it goes down again afterwards and recovers back to the 0, the baseline. But this has been a really important modality for working out all sorts of things about how the brain works. Now, at ImageX, we're kind of really focused most of our work around radiotherapy. And I think, and you guys have probably heard a lot, or have heard a lot up to this point about CT and X-ray based imaging modalities that have been used for ages in radiotherapy. So there's really sort of this question of why do we need MRI for radiotherapy? And that's because the conventional imaging techniques have terrible soft tissue contrast. X-ray techniques, CT techniques have terrible soft tissue contrast. So to give you a comparison here, we have someone with prostate cancer. And on the left, you see that prostate, this region here, has this really even grey tone across the whole thing. On the right-hand side, we have an MRI zoomed in of the same prostate, and you can see that we get all this amazing structure that actually also just shows the tumour here at location as well. And that's because T1 and T2 in these two different tissues are different between the tumour and the healthy prostate. And we can turn that into an image here. Now, what one of the real sort of developments in the last few years and what we work on here at ImageX is the idea of this MRI linac, or it's an MRI system combined with a linear accelerator. And so what these MRI linacs do is they image the person in real time as X-ray radiation therapy is delivered. And so in this case, this is a schematic of our prototype MRI scanner that we have at Liverpool Hospital. And we have a number of projects that are running across this. So I thought I'd sort of run you through in very high level detail about some of the things we're working on. So to jump into that, so at ImageX, we've got some of you may have met Dr. Paul Liu, who's been sort of really leading the work that we've been doing on using MR imaging to dynamically adapt a radiotherapy beam. So for those of you that, and I know some of you have been in the clinic too, what we have here is a phantom, a moving phantom that's mimicking lung motion, moving a hypothetical tumour. Here we're using the MRI scanner to image those moving tumours in real time. And over here we have the MLC pattern, or the multi-leaf collimator leaf pattern being sent across to track the tumour, to track the tumours. And here we have the actual radiation that's being delivered to this phantom as a result of the imaging. And you can see here that what we're able to do is track these tumours in real time. And that's really important if we want to minimize the dose to to tissue around moving tumors. We really don't want to be just spraying radiation dose everywhere in the hope of hitting a tumor. We really want to be able to track it. So another project that we have going, and for the people working on these, I've just got it very broadly. My project is actually one of my main projects is based around the idea of integrating neural networks into MRI scanners. And so I spoke earlier about this idea of K-space and filling up K-space. And all I'll say is that neural networks have done lots of amazing things. They've also proved that they are amazing and better than usual methods of turning that K-space data off the MRI scanner into an image. And they can do it really quickly. So one of the things that we've been implementing on our scanner is the actual using these networks to perform that operation and track moving tumors or moving phantoms, tumor-like behaviors. And so Sony from the summer student cohort this year has been working with me on that, and James Grover has been really important to that effort as well. So we also have an effort led by Dr. Shen on improving image distortion. So as you can imagine, when in radiotherapy, when you're wanting to target this tumor, geometric precision is crucial to whether or not you hit the tumor or not. And Shen has a whole suite of techniques, and I've just sort of shown a demonstration here for correcting what can be pretty dramatic image distortion and turning it into a nice image. So in this case, we have a phantom that is nice and square, ready to be imaged. And you can see it's all wobbly in the original one and turns into a beautiful image here that's better than conventional methods here, which is shown in the middle. The simple harmonic method is the SH. So another project we have is looking at using, led by Dr. Katarina Briggi, looking at using multiparametric MRI imaging to more accurately target radiotherapy with MRI. So the idea here is that, well, for a start, brain cancer is a terrible disease that kills more people under 40 than any other cancer. And one of the main reasons for that is it's just a nightmare to work out where it really is and where it's going to recur and where it's going to be. And so the idea of this project is actually to use all these differences in T1 and T2 to generate probability maps of where the tumor is, where it will be, where it might recur. And then the idea is to turn that into dose boosting plans that deliver more radiation dose with radiotherapy to regions where to regions where we think a tumor might be. So, and another sort of project that sort of just falls under the MRI Linac remit is led by Dr. Brendan Whelan. And this one is really not so much about the MRI imaging itself, but more about the fact that moving metal in magnets, so the way that we normally reshape radiotherapy beams is by shake our radiotherapy beams with these multi-leaf collimators. And it turns out moving lots of metal in a magnetic field is not always the best idea. And so Brendan has this thing that I like to think of as an old school cathode ray television that he calls the Sphinx, that instead of moving metal to reshape a radio beam, actually uses a collimator and this electron gun to steer. Did my screen go crazy for everyone else then, right then?
Yes.
Yes.
That is wild. I think that's, okay, cool. I think fixed. Yeah, anyway, and so that's sort of, yeah, another bit of research that we have going on there. But otherwise, yeah, that's some of the things we're working on. I hope that people remember T1, T2, TR, TE, the 4 characteristic times that give you an image in MRI. And thank you for your attention, and happy to take questions.
Thank you, David. Does anybody have questions for David? All right. I might jump ahead and ask you a question, David. Do you know, like you mentioned, there's like T1, T2 images, and they can highlight different tissues. Do you know if there's like combined T1, T2 images? Is that something that's also being done, or is it?
So yeah, so there are entire books of just like all the different types of images you can take and all the different variations. So really what those terms of T1 weighted images and T2 weighted images are really more like clinical practice than anything intrinsic to the way that the image actually occurs. And so it's more that clinicians expect to see an image to look a certain way. So all images that you acquire will have some sort of signal intensity that is, and difference between tissues that is some combination of the T1 and T2 properties of that tissue. You tune the parameter, the TR, the TE on the MRI scanner so that you get an image where the contrast is dominated by the T1 effects or it's dominated by the T2 effects. But there are also, so like one of the issues that we actually have on our MRI Linac is that typically actually the real-time imaging sequences give you contrast that is T2 divided by T1. And that can actually kind of make it much more challenging to tell the difference between some tissues.
Cool.
They tend to scale together.
So I have a question, David.
Yep.
So with multi-parametric effort to actually track the tumor, like how big is the blood flow to the tumors and related to the other cells around? Like, do you have like a huge contribution from the tumor?
How do you mean? So like, I guess what the like in terms of the relative cerebral blood flow, relative cerebral blood volume, those differences are significant, but the problem is trying to work out that it's to do with that specific tumor and not other things that occur in somebody that has a brain tumor. So there's all sorts of edema due to swelling in sort of in cancer patients, for example. And yeah, I mean, I think, yeah, so I think it's more that the differences are there. The question is, I think more so, how do you know that it's due to tumor and not something else? And I guess also Kat would also be able to give a much more detailed answer than me to that.
Unless you have a task-dependent effect there, you won't really able to say anything about it.
In terms of like what is significant?
Yeah.
All right. Anybody else have a question for Brendan?
Or David?
Or David? Sorry. For David. All right. I have a question for you, but I'm not sure if it's your area of expertise so much. But I know MRI Linac. So in a Linac, when you deposit those, it's X-rays that become electrons. So if you're doing that in a magnetic field, there are some effects that occur. So do you know whether it's advantageous or does it make it more difficult to plan?
So yeah, so I mean, I guess the idea is that you have the ionizing X-ray, and then you have the electrons that are generated, and then your electrons, rather than just going straight in whatever direction they would normally do, because they're in a magnetic field, they can go in cyclotron orbits and get stuck. There is some stuff that some people, including people in ImageX, have sort of looked at, that you possibly get some small enhancement of the dose as a result of that, the fact that just the path lengths are longer. I think my understanding is, and I think Hillary is maybe a better person to ask this to, my understanding is it's not that significant. Like it's a couple of percent, a few percent, maybe it's not 50%.
All right, cool. And then if no one else has any other question, I have one last, which is I know that the MRI Linac is like the Australian MRI Linac has a lower field strength than regular imaging MRI. Does that create challenges when it comes to tracking tumors or organs at risk, or is it not really, like, is it a non-issue?
Yeah, interesting question. So I think what it has, so there's two commercial systems that are really out there at the moment. There are other systems in development, including our one. There's the Unity Elector system, which is made in collaboration with Philips, and then there's the ViewRay Meridian system, which is based on a Siemens MRI scanner. But so you have these two things, and the Unity Elector system is 1.5 Tesla, the same as traditionally most MRI scanners, diagnostic MRI scanners. And then you have this 0.35 Tesla ViewRay Meridian system, which is around the 0.35 T mark from memory, but it's low, as you say. The difference in field strength for the quality of imaging you need for tumor tracking, I think, in my opinion, is negligible. But the impact on sales and marketing to clinicians has been enormous. So even though in sort of my opinion, the ViewRay has been technologically a long way ahead of the Unity Elector system. So they were, for example, they implemented at least a couple of years before Unity did a gated system for stopping the beam with significant tumor motion. They're still selling nowhere near as well as the Unity Elector system. And generally, as far as we can tell, that's down to the fact that clinicians are like, oh, well, I want a good MRI scanner versus, even though they're a lot more expensive.
That makes sense. All right.
I also have a basic question. So with this Shunshan's distortion correction work, like if you do not correct for it, what will happen to what are the improvements that you expect from that? Is it like in the tracking accuracy and?
Well, I guess it's, basically, so I guess it's a problem that is much larger on an MRI Linac than it is on a standard MRI scanner. Mm-hmm. So on a standard MRI scanner, the whole system is designed for high quality diagnostic MRI. Whereas on the MRI Linac, typically you have this gantry that has the X-rays coming out of it, and that's a giant chunk of steel that is distorting the magnetic field. Mm-hmm. So basically, it's really comes down to a question of how accurate does your radiotherapy need to be, but essentially that effort is about being able to tolerate much larger image distortions and correct for them so that you're guiding based on the best image.
Yeah. Oh, thanks.
All right. Any last question for David? All right. Thanks, David, for the lecture. It was really interesting.
Thank you.
Anybody who wants an attendance certificate, please email Julia. Her email is in the chat. And yeah, I will leave the Zoom open for a couple minutes. for you to copy the email or ask David questions, and then I will give you a notice of about 1 minute before I turn off the Zoom.